The Joint Clinical Research Center is an international center of excellence that was set up in 1991 by His Excellency Yoweri Kaguta Museveni to find a scientific intervention in the HIV epidemic. The work of the center has extended beyond HIV to address the COVID pandemic that is ravaging the world today.
The center is among the first 12 laboratories that were approved to support Ministry of Health in COVID-19 testing, a service it has offered since 2020.
The tests for doing COVID testing are majorly imported into the country, and this makes the cost of the test very expensive, at about $65 a test. Because of the high cost, the government has prioritized groups to be tested, not everyone. And this has also limited the community monitoring of the spread of COVID because of the cost. Majorly, samples are collected and centralized at high-level laboratories like this at JCRC, which use complicated equipment, and the processes for testing are long.
Testing is a major pillar in the COVID-19 pandemic. Therefore, scientists at the Joint Clinical Research Center are employing an already existing viral load platform, and they are using it to develop a COVID-19 diagnostic platform that is affordable, high throughput, easy to use, with readily available reagents that can be used as a point-of-care platform in centralized high-end laboratories as well as rural facilities.
The gold standard test for COVID mainly has two steps, and these have been modified by JCRC to create 3 platforms. These 3 platforms include the RARA, which is mainly used for routine testing of COVID and disease management. The other 2 are mainly used for mass screening of COVID infections in communities so as to control or manage the epidemic.
The assays are a dramatic simplification over the traditional PCR test methods, taking about 30 minutes compared to the 7.5 hours it takes on the conventional PCR platform. The device I'm holding in my hand takes about 2 minutes to extract 96 samples compared to 4 hours on the conventional Abbott platform, while the second stage on the conventional Abbott platform takes 3 hours compared to 30 minutes on my new assay.
The proof of concept for the RARA assay has showed that COVID-19 diagnosis is possible. The assay uses both the saliva sample and the nasal swab that is currently collected. We have done over 1,000 tests using this assay, and it has shown a very high performance. The assay has transitioned from the manual platform to the automated equipment. The equipment runs many more tests per day. The automated equipment has been delivered and installed at JCRC, and training has been completed.
We are in advanced stages of launching the commercialization of the assay, working with National Drug Authority. The kits for the test will be assembled locally and provided for routine testing, and local production of some of the reagents is already in progress, including 3D printing of some of the plastic supplies that are traditionally imported into the country.
The other 2 assays we have developed will be used for community mass testing to monitor the epidemic within the communities and to detect emergence of new strains. For this, we will use more sophisticated and more advanced next-generation sequencing approaches. This has the ability to detect and to monitor evolution of the virus within the community, even when present in very low levels. Equipment for this has already been obtained.
So today the government of Uganda is asking Ugandans to get the various COVID-19 vaccines to stimulate the body to produce antibodies or soldiers to fight against the COVID-19 virus. This is called active vaccination. And once your person is vaccinated, it takes about 2 or so weeks for you to develop these antibodies or soldiers. However, there is also what is called passive immunization, where there is transfer of antibodies from somebody who has been infected by a disease to a patient, either to protect that patient or to treat that patient. Convalescent plasma, or the fluid component of our blood, is collected from patients who have recovered from a disease, and this convalescent plasma has high levels of antibodies that can be transferred to a patient for treatment or for protection against disease. This way of treatment was used in the treatment of Ebola during the Ebola outbreak recently. COVID-19 convalescent plasma has been used extensively in China, Europe, UK, and the US during the first and second waves of the epidemic. And what emerged was that for patients who presented early, if given convalescent plasma with high levels of antibodies, this prevented severe disease, hospitalization, and death. To collect convalescent plasma, we use a machine, an apheresis machine called a trimmer machine, and we use these kits that are connected to this machine. And for each patient, we use a single kit. As you can see, the kit is well sealed, and the kit inside here attaches to this area.
USA has gone further to concentrate the antibodies from plasma and is planning for a large study of over 2,000 COVID-19 patients using plasma that has been collected from recovered American patients. Uganda needs to be prepared to manage COVID-19 patients and prevent severe disease in case we get a third wave with as many options as possible. So JCRC scientists have built on the existing facilities to collect plasma and prepare concentrated antibodies using plasma that has been got from these recovered patients. And we are storing it so that it can be used in case it's required.
We have identified eligible donors who have recovered from COVID, who will be able to donate the plasma for prevention and treatment of COVID. The equipment has been installed and training has happened, and we've also purchased kits to cover about 1,000 collections of plasma. The study guidelines are undergoing regulatory review in the country, and as soon as we have approval to start, we will collect the plasma, concentrate the antibodies, and store them for eventual use if we have any third wave of COVID that comes up. So we should be prepared when we have a surge of cases of COVID to treat them.
Storage space for plasma and concentrated antibodies has been prepared. Monitoring of temperature is done 24 hours using automatic data loggers, and we have a backup generator to ensure that there is no cut-off power from the freezer house.
So in addition, the center received a new equipment called the Miraso. This equipment will provide the further additional safety to the blood products that we give the patients by killing any organisms that could be present in the blood.
These two research projects have been implemented at JCRC, enabled by funding from Government of Uganda through PRESIDE. This work has been done by scientists at JCRC who have worked tirelessly to address the local problem. We are grateful to the Government of Uganda for being supported to find local solutions that have great potential to impact the economy of Uganda.